In this video, we are going to understand how cells synthesize the building blocks of DNA called deoxyribonucleotides. These molecules are absolutely essential because without them, DNA replication cannot occur. Think of DNA like a sentence. If even one letter is missing, you cannot complete the sentence. Similarly, without all four DNTPs, DNA replication will stop. So how cell synthesizes DNTPs, we are going to study it in this video. As we know that DNA synthesis requires four precursors which are DATP, DCTP, DGTP and DTTP. These are collectively called deoxyribonucleoside triphosphates, and these are derived from RNA precursors which is ADP, GDP, CDP and UDP, and they are converted into their respective deoxyribonucleoside triphosphates. How ribonucleotide is converted into deoxyribonucleotide? In this figure you can see ribonucleotide, that is precursor of RNA or building block of RNA. It is converted into deoxyribonucleotide, the building block of DNA, with the help of an enzyme, that is ribonucleotide reductase. How ribonucleotide reductase carry out this reaction of a ribonucleotide conversion to deoxyribonucleotide can be understood in this slide. Here you can see on 2' carbon there is OH group, and here on the 2' carbon there is H, hydrogen atom only. It means this is ribonucleotide and this is deoxyribonucleotide. Ribonucleotide reductase has removed this OH group and it has added this hydrogen atom, and water is formed as a byproduct of the reaction. So when ribonucleotide reductase provides its hydrogen atom to the 2' carbon of the ribose sugar, it is oxidized. After providing its hydrogen, this enzyme is oxidized. In oxidized state, the enzyme is inactive. So enzyme should be activated to carry out further reactions to convert ribonucleotide into deoxyribonucleotide. Here is the step where thioreduction comes into play. It provides its hydrogen to the ribonucleotide reductase, and this thioreduction is also converted into thioreduction in an oxidized state. It means here it will be inactive, so it also should be activated so that it may continually supply the hydrogen atom to ribonucleotide reductase. So this thioreduction gets electron or hydrogen atom from the thioreduction reductase enzyme. This enzyme gets hydrogen from NADPH plus H plus. It converts NADPH plus H plus to NADP plus. So the hydrogens are transferred from NADP H plus H plus to thioreduction, and from thioreduction they are transferred to the ribonucleotide reductase, and from ribonucleotide reductase electrons or hydrogen atom are transferred to the ribose sugar at 2' carbon so that it becomes deoxyribose sugar. Here you can see the conversion pathway of ribonucleotide into deoxyribonucleotide. The first step is the reduction step and the second step is phosphorylation, in which a phosphate group is added to make diphosphate into triphosphate. But at the end you can see here DUDP is converted into DUTP. Do we need DUTP for the DNA synthesis or DTTP? Obviously DTTP. So DTTP is synthesized from DUTP in a process which will be discussed in the further slides. DTTP is synthesized by a special pathway in which UTP is converted into DUTP with the help of ribonucleotide reductase. DUTP is converted into DUTP with the help of kinase which will add a phosphate. DUTP is converted into DUMP with the help of phosphatase enzyme. It will remove two phosphates. Then DUMP is converted into DTMP, and DTMP is converted into DTTP with the help of kinase enzyme which will add phosphate group. So what is the difference between uracil and thymine? You can see in this figure, this is uracil and this is thymine. The only difference between thymine and uracil is that thymine has methyl group on the 5 prime carbon. Mean thymine is actually uracil, but it is methylated uracil. Here we can understand the step-by-step synthesis of deoxythymidine triphosphate. In the first step, you can see uridine diphosphate is converted into deoxyuridine diphosphate, and it is mentioned here that ribonucleotide reductase converts ribose to deoxyribose. In the second step, phosphate is added to make it deoxyuridine triphosphate. So now phosphatase enzyme will act upon the DUTP and it will remove two phosphates and it will be converted into DUMP. So phosphatase removes two phosphates. In the third step, there is conversion of DUMP to DTMP. Here, a methyl group is added by tetrahydrofolate to DUMP so that it becomes DTMP, and tetrahydrofolate is converted after giving a methyl group, it is converted into dihydrofolate. So third step is mentioned also here that thymidylate synthetase uses THF tetrahydrofolate to donate a methyl group. So tetrahydrofolate is converted into dihydrofolate. In the fourth step, kinase enzyme adds a phosphate so that DTMP is converted into DTTP, mean triphosphate. So kinase adds two phosphates to form the triphosphate, and the tetrahydrofolate was converted into dihydrofolate. It will be converted again into tetrahydrofolate. So the enzymes are mentioned at the end of the slide. First is ribonucleotide reductase, then kinase, then phosphatase, thymidylate synthetase. What is the role of thymidylate synthetase? It adds a methyl group from tetrahydrofolate to the uridine monophosphate, deoxyuridine monophosphate. And then at the end is dihydrofolate reductase, which will convert the dihydrofolate back to the tetrahydrofolate. Thanks for watching, like and subscribe.